Hello everybody, my name is Elliot Kimmel and this is my monohybrid problems tutorial video. And in order to get the best out of this video, I'm going to suggest that you get the monohybrid problems tutorial worksheet that I'm going to show you about here. It's available on my website, www.zerobio.com. You go to the Student Center and then you go to the grade 11 course pages and then scroll down to the genetic processes worksheets strand, and you can find the printout. So I'm going to show you that right now. Here's my website, Zerobio, and we're going to go up to the Student Center and then the grade 11 biology course. And here's the genetic processes strand. Click on the worksheets and that'll take you to all the stuff that I've got associated with this strand here. And scroll down to monohybrid problems tutorial worksheet, the one that says use with video. And it's an Adobe Acrobat PDF. So you just click on that and that will load the worksheet that I've created to go along with this video. Now, what I would suggest to you is if you've got some genetics background, you could just go ahead and try the problems on your own and then watch the video to see the solutions, or you can work on it with me. All right, so that's how to get the worksheet. And now let's have a look at the actual video tutorial. So I'm going to be looking at 5 different genetics problems, pretty standard monohybrid problems. We're going to look at tongue rolling, freckles, eye color, the dash technique, what I call the dash technique anyhow, and test cross. All right, so here we go. Here's problem 1. So we've got two chimpanzees are sticking their tongues out at each other. If the male chimpanzee can roll his tongue, which is a dominant trait, but the female chimpanzee cannot, and they have babies, will any of their offspring be able to roll their tongues? What are the phenotype and genotype ratios for the offspring? Note that the male chimpanzee is heterozygous for tongue rolling. So let's go through a little bit of the theory first as an introduction to this. I want to talk about this heterozygous concept right here. If we've got 2 genes, let's say you've got big T and big T, this is called homozygous when there's 2 of the same genes. When you've got little t, this is also homozygous, but this is the homozygous recessive and this is the homozygous dominant. Heterozygous means you've got 2 different genes. All right, they're both coding for the same trait. In this case, it's going to be tongue rolling, but there are 2 different size genes. So we've got the dominant gene here, and we've got the recessive gene there. So that's called heterozygous. So knowing that, we can go ahead and start working on the problem. Now, the first thing to do is to define our genes. We're talking about tongue rolling, and being able to roll your tongue, or a chimpanzee being able to roll its tongue, is a dominant trait. So I'm going to choose capital T to represent the rolling trait. Therefore, little t will represent the gene that causes non-rolling. So now we're going to work on the cross. For all of these problems, I'm going to put the first individual here, the first individual mentioned in the question in the first spot, second individual in the second spot. So this male chimpanzee is heterozygous, so that must mean it's going to be big T, little t. So if I uncover this, big T, little t. Now, as you're working through the worksheet, you just go ahead and write down the answers with me, or again, try it ahead of time and then come back and see what I do. The second chimpanzee is the female chimpanzee. She cannot roll her tongue, so she has to be little t, little t. That's the only way. Now notice he is heterozygous and she is homozygous recessive. Now it's time to do the Punnett square. Now, when I do this, I like to put the first individual over on the side and the second individual I like to put in the top here. Now, it's not absolutely necessary, but that's the way I do it. So we're going to take these genes and we're going to split them here. This is going to be a big T and this is going to be a little t. Now, will you please notice something important here? This male chimpanzee has 2 genes coding for the tongue rolling trait. And this is because there is one of these genes on one homologous chromosome, this one, and the other gene on the other homologous chromosome. Let's say this is chromosome pair number 1. And there are a lot of genes. If this is the chromosome, there's genes here and here and here and here, all the way along. But at some point is the gene causing the big T trait. And on the other homologous chromosome, there are also genes, all right? And this is where the little t would be. So these are homologous chromosomes, and this is the diploid condition, where there are two chromosomes or two genes coding for each trait. But in the Punnett square, I've separated them out. I put one big T here and one little t here, and that's because these represent the gametes with half the number of chromosomes. So if there were 46 chromosomes up here for a human, let's say, there would be 23 chromosomes down here. So there is one gene in each spot representing the haploid condition. Same thing goes for the female. Up here, she's going to be little t, all right, haploid gametes with all of the other genes, but right now we're doing monohybrid problems, so we're only focusing on one trait, one gene. Now, we are going to take the first big T and we're going to cross it with the first little t, and we're going to put the result down here. And we are going to reveal that. We'll get big T, little t. This represents one of the offspring. If this was a sperm from the male chimpanzee carrying the big T, and this was an egg from the female chimpanzee carrying the little t, then when they united in fertilization, the baby would have big T, little t, along with all of the other genes. Another possibility is this big T with this little t, so we get that. And then we take the bottom one here, this little t from the male chimpanzee, and this little t from the female, and we get little t, or this one with this one, little t. And that is the completed Punnett square. Now, we need to answer the first question, which is, will any of their offspring be able to roll their tongues? Because tongue rolling is a dominant trait, we can see that this chimpanzee and this one both have a big T. Doesn't matter about the little t. This little t is recessive and it's masked by the dominant gene. So this one can roll its tongue. This one can roll its tongue. But this one with two little t's cannot. This one cannot. So we can go ahead and answer the question. Yes, half of the babies will be able to roll their tongues. These ones right here. Now, phenotype ratio. The term phenotype refers to the outward expression of the underlying genes. So the genes here, like big T, little t, this causes a trait, right? The phenotype for this chimpanzee is tongue rolling. The phenotype for this chimpanzee is non-tongue rolling. So we're not talking about the letters or the genes, but we're using them to figure out, what the organism is going to be able to do, what the trait is. So the phenotype ratio is going to be a ratio between tongue rollers and non-tongue rollers. So here we have two of them that can roll their tongues. We have two of them cannot roll their tongues, and we're going to bring this down to a 1-to-1 ratio. So that means there is one roller, this one, to one non-roller. Well, there's 2-to-2, but we bring it down to lowest terms, 1-to-1. The genotype ratio, well, genotype refers to the actual letters that cause the trait. So here we can see big T, little t, that's called a genotype. The phenotype would be tongue rolling. The genotype would be the actual letters, big T, little t. All right, and as you can see, there are two of these, two big T, little t's, and two little t's. So this becomes, again, a 1-to-1 ratio of traits or genotypes. So that's one big T, little t to one little t. We've solved problem 1, and it's time to move on to problem 2. Freckles is a dominant trait in humans. Freckled parents have 4 children, and 3 of them have freckles. Determine all genotypes to complete the cross in Punnett square. Are any of the children homozygous for freckles? Give the phenotype and genotype ratios for the children. All right, so freckles is dominant. Therefore, in order to do the cross, we're going to define our genes again as usual. I'm going to put a capital F here for freckles because it's the dominant trait, and a little f for no freckles because it's the recessive gene causing the recessive trait. Now, we don't know enough to do the cross right now. All we know is that the parents have freckles, all right? But we don't know if it's big F, or big F, little f. We're going to figure that out from the other information in the question. We've got 4 children that they produce, and fortunately there are 4 in the Punnett square, so it makes it easier. 3 of them have freckles. Now that's the dominant trait, right? Non-freckled is the recessive. So 1 of them does not have freckles. And usually when you have more with a particular phenotype than the other. I didn't say that very well, but the other, in this case, goes down in the bottom right corner. So this is the individual that does not have freckles. This is homozygous recessive, as you can see, 2 little f's, does not have freckles. The other 3 will have freckles. But these F's had to come from the parents, right? This F came from here. This little f came from here. So now we know something about the parents. They had to donate those genes to produce the offspring. So we know that both parents are carrying the recessive gene for freckles. So therefore, because the parents do have freckles, as we're told in the question, the other gene for each parent must be a big F, because they have freckles, but they have a child that doesn't have freckles. So they must have contributed that gene. Well, now we can figure out the cross. Big F and big F right here. Big F and little f right here. And I like to switch around the dominant and the recessive gene. So instead of writing little f, big F, I'll have that as big F, little f. All right. Now we can go ahead and reveal the cross. We've got one parent that is heterozygous for freckles. The other parent also heterozygous for freckles. So we'll have big F, little f for the first one, big F, little f for the second one. Okay, now let's answer the question. Are any of the children homozygous for freckles? Well, can you see that? This is the one that's homozygous for freckles. Homozygous dominant, right? 2 of the same. This one is homozygous, but it doesn't have freckles. So that doesn't answer our question. So, homozygous for freckles? Yes, 1 child, this one right here. Let's look now at the phenotype ratio. This one has freckles. This one has freckles, this one has freckles, this one doesn't. So it's going to be a 3-to-1 ratio of freckles to non-freckles. 3 freckles to 1 no freckles. Let's do the genotype ratio. There is one of these big F's, homozygous dominant, but there are 2 big F, little f's. Now that's really why I switched this one around so it was the dominant before the recessive. It makes it easier to see that these 2 are the same. And then there is one more of these. So there's 1 to 2 to 1. Again, we're looking at genotype ratio, so we're actually looking at the letters. So 1 big F to 2 big F, little f's to 1 little f, little f. And we have solved problem 2. Problem 3. My wife has dark-colored eyes. They're black, which is the dominant trait. All of her family and her relatives have dark eyes as well. So what I'm getting at in this question is the fact that all of the individuals in her family and all of her relatives, they all have the same eye trait, okay, dark eyes. And that essentially will make my wife's eyes, the dark eyes, homozygous for that trait. I, on the other hand, as you might be able to see, have colored eyes, light colored eyes, they're blue, which is the recessive trait. But we are hoping for a blue-eyed baby. What is the probability we will have a baby with blue eyes, and what is the probability we will have a baby with dark eyes, brown or black or something like that? Well, because dark eyes is dominant, we're going to put a capital E there to represent the dominant gene causing the dominant trait. Light eyes is recessive, so my blue eyes is caused by little e. Now, let's do the cross. Because I said that all of her family and relatives have dark eyes, we are going to make the reasonable assumption that she is homozygous for that dominant trait. So she's going to be big E, big E. And because I have light eyes, blue eyes, I am going to be little e, little e. So we can go ahead and do the cross, putting the first individual on the left side, big E and big E, and me up here, little e, and then we'll cross them. This one times this one, big E, little e. This one times this one, big E, little e. Do the same for the bottom row, big E, little e. And this one times this one, big E, little e. All right, we've got the cross and we've got the Punnett square. We can now answer the question. Can my wife with her dark eyes, homozygous dark, and me with my blue eyes, can we produce a blue-eyed baby? No, we cannot. Probability is 0% of a blue-eyed child. There's no little e possible here. What is the probability of having a baby with dark eyes? Well, it's going to be 100%, right? All of them are going to have dark eyes. Good. On to problem 4. A blue-eyed woman and a brown-eyed man have children. If the eye color of the man's family and relatives, and therefore his genotype, is not fully known, determine the phenotype and genotype ratios for their potential children with respect to eye color. So we know something about the woman because she has blue eyes. So this, of course, is going to be caused by little e. And, of course, the dark eyes will be caused by capital E, which causes the dominant trait of dark eyes, brown eyes being dark. But we don't know enough about his family and his relatives, so we can't make the assumption, like we did in the previous question, that he is homozygous dominant for that big E, all right? We know he's got at least one big E, but he could be big E, or big E, little e. We don't know that second gene. And so what we're going to do is, in the cross, put down that this is the woman, sorry. The woman with blue eyes is going to be there. And for him, because he comes second in the question, so I'm putting him second there. I forgot that. We know he's going to have a capital E, but we don't know the other gene for sure. So I'm going to use a dash. All right. You could use a question mark or an X or whatever you want as a placeholder for this unknown gene. I like to use a dash. So we're going to put these into the Punnett square, and we'll have little e, and I see I've already got one revealed there, and little e and the dash. So when they cross, the big E and the little e will get together. Again, I like to switch the order of these, put the big E first before the little e. And then when we cross this little e with the dash, we are actually going to put the dash into the Punnett square. We're going to take these two and cross them, putting the dominant gene first, and then little e and dash. Now, this is our completed square, but admittedly, there's some unknowns here, all right? We don't know what this dash is. So when we look at the phenotype ratio, keep in mind that this dash could be a big E, right? And that would have made him big E, having homozygous for dark eyes. If this dash is a big E, then this becomes a big E, this becomes a big E, and now all of these have the capital E, the dominant E gene, and therefore all of the individuals are going to be with dark eyes. So to do the phenotype ratio, we have to consider this. We say if the dash is big E, then what will happen? Then we're going to get 4/4 or 100% with dark eyes, or brown eyes in this case, because we're wondering if they have brown eyes, all right, like the man. However, if the dash was a little e, if that's a little e and this is a little e, then all of these become little e's. These two now have dark eyes, let's say brown eyes, and these two now have light eye color, and so that makes the ratio different. So we say if dash is little e, then we are going to get, for the phenotype, 2 dark eyes or brown eyes to 2 blue, or in lowest terms, 1 to 1. 1 brown eyes to 1 blue eyes in the offspring. We do the same thing when we look at the genotype ratio. We have to say, if the dash is the dominant gene, the big E, all right, then we're going to look at the actual letters. So if this is a big E, and this is a big E, and this one, what are we going to have? Well, if that's a big E, then in the genotype, all of these end up being the same. So this is 4/4 big E, little e. All right, we get 4/4 or 100% big E, little e. But if the dash is a little e in this case, okay, so now we got a little e, back to writing the same thing I did a minute ago. All right, now we're going to have two different. We're going to have these two as a distinct genotype and these two as a distinct genotype. So we are going to have a 1-to-1 ratio, 1 big E, little e to 1 little e, little e. All right, so there's some unknown. We don't actually have the final answer to this. We only can say, well, if this is the case, then that's what we'll have. And if the other thing's the case, then the other thing is what we're going to have. All right, let's do a similar problem over here with this farmer guy. A farmer wants to know if his prize cow is homozygous for black spots or heterozygous. Black spots is dominant. He has both spotted and non-spotted cattle. All right, so he's got lots of different varieties, but this particular cow, this one is special. How can he figure this out? How can he figure out if his cow is homozygous for black or heterozygous? We know that black is dominant, and so we're going to come over here and say spots is caused by big B, the dominant gene causing the dominant trait. Therefore, no spots will be caused by little b. So we don't know what the genotype of his cow is. We do know that the cow has one big B at least because it's got spots, but it could be big B, or big B, little b. So we're going to put a dash in there, our unknown, right? So whenever you don't know for sure, you can use this kind of technique. Now, it is possible to figure out what the genotype of his cow is and determining, therefore, if it's homozygous or heterozygous. It is possible to do this if you do something called a test cross, and I've written that over here. This is a test cross. We are going to test the genotype of his cow by doing an actual cross, and what we cross it with is a homozygous recessive individual. So this is the unknown here crossed with a known individual. This would be a non-spotted cattle. All right, non-spotted. And in order to be non-spotted, it has to be homozygous recessive. So he performs this cross, and then we're going to look at what the potential offspring are. So here's how we write the cross. Big B and dash for his unknown cow. Little b and little b for the known non-spotted individual, and then we cross them. This offspring is going to be heterozygous, and it's going to have spots. Heterozygous, and it's going to have spots. Now, we're going to have the dash in here. I like to put the dash second, all right? Put whatever gene we know here and put the dash second. So we're going to have this in our Punnett square. And then you say, I don't know if we're any further ahead. Well, we are, because these are real potential baby cows that the farmer can have. And we look at them and we have to say this. If the dash was a big B, so let me come up here. If the farmer's cow was big B, all right, then this dash would have been a big B, and then all of these would be big B. So what is the farmer going to see in the offspring that are born? He's going to see spots, 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 spots. You know, if he has 4, but I mean, if he has 40, if he has 400, all of them are going to have spots. If that is the case, the farmer now knows that the second gene was a big B, and this is therefore the genotype of his cow, big B. So if dash is big B, all cattle will have spots, and therefore his cow is big B. It's homozygous, so now he knows that. On the other hand, if the dash was a little b, and he doesn't know that yet, right? If it was heterozygous, this would have been a little b. So would that. So would that. And now, in the offspring, half of them would be spotted, half of them would be non-spotted. So if we had 4, potentially, by probability, 2 of them would have spots, 2 of them wouldn't. Or if he had 400, 200 have spots, 200 don't have spots. That's a lot of babies to have. All right? So we look at that scenario here. If the dash is a little b, then half of them will have spots. And if that is the case, his cow is heterozygous. So a test cross is done in order to determine the unknown genotype. You cross the unknown with a known genotype, and the only way we can really know for sure is when we have a homozygous recessive here. We cross that with the unknown, and then we look at the offspring. That's why it's a test cross. We look at the offspring, and then we can work backwards and figure out the exact genotype of the unknown parent, and that's a test cross. All right, so that's my little problem set on monohybrid problems. Hope that helped. See you again soon.
I'm ready to fall, and I'm betting all the ways of my heart. I'm ready, so ready to fly to the end of the sky on the wings of my heart. Oh, I'm ready suburb.